Aloha! Today my presentation will focus on the mechanism of DNA replication. I will begin by briefly going over the general structure of DNA polymerase III. It is a multi-subunit complex made by DNA B or helicase that is attached to the two core subunits that actually perform the synthesis of the new DNA molecule. The binding of the core subunits is guided by the beta clamps that are loaded by the clamp loading complex. Now, you will see how this works a little bit better, especially in a video that I will show you, or that I will ask you to watch. And so this would make a lot more sense when you see it, how it actually works a little bit later. This crystal structure and representation shows how the helicase associates with DNA. This is from the top, and this is from the side view, where we see the helicase associated with DNA. All right. Now, in order for replication to occur, many proteins will be involved in that process. It's been estimated that in E. coli, about 20 different enzymes and proteins are involved in the replication process. All these proteins, based on their function, can be divided in three groups: proteins and enzymes that supply the template. And it's not so much to supply the template, because the template itself is the DNA, but not the double-stranded DNA. The template is the single-stranded DNA, and something needs to make DNA to split and allow to be used as a template so that a new molecule of DNA to be synthesized, a new strand to be synthesized, a complementary strand to be synthesized. The enzymes that are involved in the supplying of the template, or the making the template available for the synthesis of new DNA strand, are the helicase that helps the unzipping, that provides the unzipping of the DNA, the separation of the two strands. Topoisomerases, which later on I will clarify, but they help to relieve the pressure in front of the replication forks. Also, we have DNA binding proteins with various functions. They either are double-stranded binding proteins or single-stranded binding proteins. But these specific ones are actually single-stranded binding proteins for the most part. The next group is the protein or enzyme that provides the primer, or the initiation site, or where replication will begin. That enzyme is called the primase. And the last group of proteins are those that synthesize the DNA copy. These are DNA polymerases that are involved in the synthesis and repair of DNA, and also DNA ligase that is involved in the repair of the nicks between the different Okazaki fragments primarily, but any other nicks that require a ligation of DNA. The stages of DNA replication are as follows: initiation, elongation, and termination. Alright, so let's begin with the initiation of DNA replication. Replication begins at the origin of replication site, which in bacteria is known as the ORI or ORIC. In E. coli, the origin of replication comprises or includes about 245 base pairs that contain several consensus sequences, such as the R sites, the I sites, and the DNA unwinding element, DUE. which is somewhat upstream of the R and the I sides of the origin of replication. Now you may ask, well, what are these sites and why there are multiple of them? Well, the multiple suggests that there are multiple proteins with similar functions or identical function are binding within that segment of DNA. And indeed, the R sites are bound by DNA A proteins, and we'll talk about them a little bit later. And the I sites are bound by DNA A proteins that are associated with ATP. The DUE element, something very specific and characteristic is observed in that element, and that's why it's called a DUE. And that is, if you look at the sequence, for example, in this particular case we see 13 nucleotides, and 9 of those nucleotides are T's or an A. 2 of the remaining nucleotides, remaining 4, can be any nucleotide, including another T or an A. So, if those ends are T or an A, that means that the 13 base pairs involved in this area, 11 would be either an A or a T. That is important. And the key of that importance is in the number of hydrogen bonds formed between the A's and T bases. As you know, A and T associates with the help of 2 hydrogen bonds, while the G's and C's associate with the help of 3, or they form 3 hydrogen bonds. In other words, segments of the DNA that is rich on A's and T's will be much easier, or at least easier to dissociate compared to a segment that is more rich on C's and G's. And we'll see that in a moment. Why is that important? How does this play a role? Now, what does consensus means? Consensus means that these sites, for example, all the R sites or all the I sites or the DUE elements, not only within the bacterial strain, but also between all bacteria, they will be somewhat similar. They will not be identical, but they will be somewhat similar. Why is that important? I mean, if you think about it, the replication process is fairly well conserved or similar between all bacteria or even higher organisms. It is natural then that the proteins that will be required for that process to occur will be similar as well, and they will associate with binding sites that have to be similar as well. So for that reason, we don't have identical, we don't use the term identical usually because there's variations between, but there are certain parts of these sequences that are very much the same or very similar compared to the same site within the same origin or the same sites within other origins of other bacterial strains. So that's what consensus means, meaning preserved. A lot of it is conserved. Most of the sequence is similar between these sites, not just within a single type of bacteria, but between all bacteria. And that is because it's the same process. All right. So that was, that's just in general the structure of the origin of replication. Now, the initiation. How does it start? How does replication begin? Well, the origin of replication, the R and the I sites, like I said, they're binding sites for DNA A proteins with and without ATP. So the DNA proteins associate with the sequence. But in addition to that, because they are placed in a very close proximity to each other, as you can see here, the R and the I sites are really close to each other. They not only interact with the sites, but they also start to interact with themselves. And when they start interacting with themselves, they start to twist the DNA molecule, adding a certain torsion within it. That torsion within the DNA molecule adds pressure on the DUE element. And as you know, this is a rich element on A's and C's. And at some point, that element will separate. The two strands in that element will separate and will form single-stranded strands. Now, once the single strands form, then the helicase, or DNA B, gets delivered and binds to the lagging strand on each of the two replication forks. DNA C bound to ATP delivers DNA B to the lagging strand of both forks. Then DNA C separates after ATP is hydrolyzed, and the helicases now remain associated with the lagging strand on each of the two replication forks. Now, we have the helicase, and once we have the helicase, that protein will recruit the rest of the DNA polymerase. After that happens, the DNA A proteins, especially those that hold ATP, the ATP will hydrolyze, and the proteins will start detaching from the double-stranded DNA. The complex that they form will fall apart, and now the replication is ready to proceed. This process is controlled by DNA methylation. The initiation of DNA replication is controlled by DNA methylation. Alright, now we've initiated it. We have the helicases associated, we've recruited the polymerases, we're about to start. Well, in order for that start to happen, we need, or the cell needs to generate a primer, which is done by the enzyme primase, or DNA G. It synthesizes a short RNA primer, short, something between 10 and 60 nucleotides. You can also see how the single-stranded areas of the DNA are bound by the single-stranded binding proteins. Now, with the helicase moving downstream on both ends, you can imagine that the leading strand will be continuously synthesized. I'll clarify this a little bit later. But the lagging strand, again, remember, we always read the template 5' to 3', but we 3' to 5', but we always synthesize new DNA from 5' to 3'. In other words, the new nucleotides are always added on the 3' end. So the leading strand will be synthesized continuously. As you can see, the lagging, though, will require periodical start of Okazaki fragments by the primase. Now, something that I want you to realize at the very, very beginning, when the first primers are synthesized, the primers will be synthesized on the lagging strand of each of the forks. Now again, imagine this is 5' to 3', and the bottom one is 3' here, and the 5' is here. The synthesis always happens after the primer in the 3' direction, towards the 3' end. Alright, now imagine now that the first primers are synthesized and the synthesis of the new DNA is happening. The helicases keeps moving in both directions, and this strand that was started at the lagging strand of the right fork will essentially become the leading strand of the left fork. And this primer, this strand that was started at the lagging strand of the left fork, will give rise of the leading strand of the right replication fork. And you can see it how this primer 1, this continues to be expressed, even though it seems like shorter, it's actually just size difference, I guess. But this will continuously be expressed without interruption and will become the leading strand. It will be treated as the leading strand of the left replication fork, and this for the right replication fork. I hope this made sense. If you have any questions, just let me know if something else, if I can be of help to explain. On the lagging strand, like I said, leading strand continuously is expressed. Lagging strand periodically, Okazaki fragments will be started, and the replication will continue. Now here you see the topoisomerase enzyme. It's always in front of the replication fork. There's another replication fork here. There's going to be also another topoisomerase on this end. The purpose of it is like, if you can imagine, is to release pressure. Now imagine that you have two strings that are tied up at the end. And imagine that they're tied, but so they cannot really change orientation with respect to each other. And then you grab the two strings in the middle and you split them. Now, first of all, imagine that they are twisted around each other. And then imagine that you pull the two strings from the middle, pull them apart. What's going to happen at the ends of the pulled apart strands? There's going to be the twisting at the ends is going to get tighter and tighter and tighter, and at some point it's going to be so hard to pull anymore apart that it will stop. It'll hold the splitting of the two strands in the two replication forks. So something is required to cut either one or both strands so that a pressure or that supercoiling to be released or relieved. We have two types of topoisomerases. We have many isoforms, but only two types. And the type of the isoform is not described in the name. The type of the isoform is determined by how many strands the topoisomerase cuts. If the topoisomerase cuts one strand, which is the case for the topoisomerase 2, again, the 2 designates the isoform, not the type. If the topoisomerase 2, for example, cuts only one strand, which is the case in our example here, that means that this is a type 1 topoisomerase. If the topoisomerase cuts both strands of the DNA double helix, that is a type 2 DNA topoisomerase. All right, so let me show you one more time how the elongation happens actually on this schematic. So you can see right here the two core subunits. They're synthesizing DNA. This is the leading strand. This core subunit is just latched on it, and it's going to continue until the end. No variations. But the lagging strand is a problem. Here you can see how one of the core subunit is using the template which is looped around it to synthesize this Okazaki fragment, but you can also already see how the single strand has already been generated because the helicase keeps moving forward. So at some point when we have sufficient amount of single-stranded sequence, DNA primase comes in, binds to the helicase, synthesizes the complementary RNA primer. Once it's done synthesizing, then it leaves the binding site, and then the clamp loading complex loads the clamp right at where we have the newly synthesized RNA primer. When the primer is bound, when the primer is synthesized and the clamp is associated, then the clamp will recruit the core subunit once the core subunit reaches the end of the Okazaki primer and it gets detached. The clamp now will basically recruit the subunit, the core subunit that will bind to it, and then we'll continue the expression of that Okazaki fragment until the next one needs to be started. So you can see in slow motion in a way, well actually in no motion, but just in the schematic, how the synthesis of the lagging strand happens. Now, there's this wonderful video that normally I'll show in class, but if I play it right now, all you're going to be able to see is just pictures, and you're not going to be able to hear the explanations of the video. So for that reason, I'm giving you the name of the video. It's called Mechanism of Replication. There is a basic and the advanced. I'll use the advanced because it has a lot more information that is more pertinent to you. And because you're not going to be able to click on the video, and I'm not going to be able to show you in this video, I also provided the link. So unfortunately, you have to type it and look so that you can get directed to that video. Alternatively, you can go to DNALC or the DNALearningCenter.org and search for the mechanism of replication specifically for the advanced video. Like I said, it's a wonderful representation. It's really interesting to watch it, how fast everything gets processed during the replication process. All right, so imagine now that the elongation has completed. I mean, obviously the leading strand will continue on and on, but imagine now that your Okazaki fragment is completed. And you're familiar with this figure. DNA polymerase 1 comes in, replaces the RNA primer with DNA nucleotides, and then it leaves. But after it leaves this nick, and there's going to be many of those in the DNA molecule, and they need to be repaired, or basically, well, repaired, I guess is a good word. And that's going to be done by DNA ligase. But how does the DNA ligase do the ligation? Well, here's the DNA ligase, and with the help of a nucleotide, either from ATP or from NAD+, an AMP will be transferred to the enzyme, adenylating it. The adenylated enzyme now is in a way activated and can help can help to repair the DNA nick. So this is the DNA nick. You see the 3' end hydroxyl group and the 5' end phosphate group, but they are not bound, and the hydroxyl group cannot just really attack the phosphate and bind with each other. That's where the ligase comes in. The ligase is adenylated, and then it transfers that AMP to the phosphate 5' end at the nick site. Now that phosphate now will be activated because AMP is an excellent leaving group. So now the hydroxyl group can attack the phosphate, and then the AMP is released. But when that happens, we have basically closed the nick, and now there's no problem anymore, no nicks within the structure of the DNA molecule. So that is the mechanism of DNA ligase. It involves adenylation, first of the enzyme, then of the phosphate, and then the release of the AMP that results in the fusion or the ligation of the strands. Okay, so we've been through the initiation, we saw how the elongation is done. Now the end, let's see how the end will happen. The signaling, the signal for the event is given by the replication fork reaching a complex between the TUS protein, or the termination utilization subunit, and the TER site, the binding site for the TUS protein. And when the replication fork reaches that complex between the TUS and the TER site in the DNA sequence, it stops and waits until the other replication fork comes in. So obviously there's going to be a part shown here, part of the DNA that's not replicated yet. We're not exactly sure how the remaining portion of DNA gets replicated. But what we know is that it does happen. We just don't know the mechanism yet. And when it happens, the two double strands were actually going to be somewhat linked and with each other. However, with the help of topoisomerase 4, which cuts both strands of the DNA, we'll be able to separate the mother or the parent strand from the daughter strand. So you can see now, again, another example of how topoisomerases are helpful. But this one is topoisomerase 4. This is the fourth isoform. It doesn't mean that it's type 4. Because topoisomerase 4 cuts both strands, that means that it is type 2. Okay, that's enough for eukaryotic replication. I'm sorry, for prokaryotic replication, or replication that occurs in the cells, in bacterial cells. And I'm going to focus a little bit your attention to the eukaryotic replication as well. I mean, in general, it is very similar in principle, very similar. But there are obviously some differences in the sequences, and the number and the types of enzymes, and some of the mechanisms that are involved. So the origin of replication in eukaryotes, it's a lot less defined, and the higher the organism, or the higher the development of the organism is, the more undefined the origin of replication is. And we have multiple sites, and the binding areas are variable, so it's a lot more difficult to determine where does replication occur in eukaryotic cells. But there is an area that normally allows for that to happen. Perhaps, the difference between a mouse, for example, or a crab, and a human is quite a bit. And even though many of the processes and proteins are similar, there's going to be a fair amount of variability between the binding sites and the proteins involved. All right, so the origin of replication is a lot less defined. In bacteria, the nucleotide or the DNA synthesis happens with a rate of 1,000 nucleotides per second. In eukaryotes, it's about 50. That's 200 times slower. Someone might say, well, that's kind of not right. It's not right because, the mammalian genome is far bigger than a bacterial genome, and we're going to have a slow replication. Why is that the case? Well, I mean, think about the implications of an error in bacteria and an error in mammalian organisms and mammalian cells. I mean, bacteria are known that they mutate quite often, but they need to act very fast, because as soon as the bacteria sees the right condition, it starts dividing massively. Where obviously that's not the case with humans, and even though the genome is larger, the replication still needs to be fairly intact, because mutations within the mammalian genome will have dramatic consequences, or could have dramatic consequences for the life of that cell or organism. All right, DNA polymerase alpha is the primase counterpart in terms of bacteria comparisons. DNA polymerase delta is the counterpart of DNA polymerase 3, and the epsilon is believed to be a counterpart for DNA polymerase 1. The termination is done at the telomeres, and as you know, over time the telomeres get shorter and shorter. The regulation of this process is very complex. It's a science of its own. It involves cyclins and cyclin-dependent kinases that, usually through interaction and phosphorylation, guide the cell and what happens in the cells in terms of the genetic material. All right, so that's all that I'm going to mention about eukaryotic replication. This chapter also includes a lot of information on DNA repair. mutations, and recombination. All I want you to do is just read on page 993 about DNA repair, and also I want you to learn what is the difference between a mutation versus silent mutation. What is the difference? And I also want you to read what's been said on pages 103 through 105 about DNA recombination, and more specifically about homologous genetic recombination, site-specific recombination, and DNA transposition. That's something that I want you to read. It's very brief. I just want you to just generally educate yourselves on these processes. We're not going to go into deep into them. All right. With that, I said everything I wanted to say about this chapter. If you have any questions, please let me know.